For inviting me. I'm very pleased to be here remotely. I'm sorry I couldn't be there in person. It's been about, I think, 15 years since I was at Neurospin, and maybe 5 years since I've been in Paris. And of course, we've been dealing with COVID ever since. Actually, I got COVID about 3 weeks ago after not having had it for almost 3 years. So fortunately, I am completely recovered. So I hope the rest of you are healthy as well. So what I'd like to do today is, first of all, quickly review behavioral methods that have been used to study infants. I'm sure many of you are familiar with some of them. And then talk about just briefly 3 topics within language development that these behavioral methods have provided us with some quite useful information about the development of language. And then very briefly review again, you're all quite familiar with these methods that have been used to study the neural correlates of development, and especially in the language domain, and then talk about a number of studies that have been done in language development using these neural methods. And finally, talk about recent work that has used naturalistic viewing paradigms, particularly movie watching, as a way to understand a little bit better how these neural mechanisms are implemented in language comprehension. So first of all, reviewing behavioral methods, it's, I think, clear over the past 50 years that almost everything we know about psychological development in infancy has been based on behavioral responses. And we have learned a lot about many different domains, such as facial expressions, learning, reaching and grasping, crawling and walking. And what drove this increase in our knowledge about many of these domains was a particular kind of paradigm that was developed that involved looking time measures. And this is just a summary article that I wrote a number of years ago describing the advantages and disadvantages of these looking time measures. And these looking time paradigms have been used in many different content domains. For example, the preferences that infants spontaneously exhibit toward different types of stimuli, effects of habituation, and the novelty and familiarity effects that we observe in habituation paradigms, the learning that occurs over the course of a short period of time in a laboratory setting, either reward-based learning or just passive exposure. And also looking gives us a source of information about what infants are naturally looking at in their environment. Now, in these looking time measures have been very useful in terms of defining sensory thresholds, such as visual acuity and cross-modality integration, highly useful in terms of perceptual discrimination and categorization, for example, in the speech domain and in recognition of faces, and also implicit knowledge that infants have about objects and events, language structure, and social interactions, such as theory of mind. So these behavioral methods, of course, have been used in many different domains, but also in particular in studies of language development. And I just wanted to review briefly some of the kind of key findings in language development that have been learned through these behavioral methods. So one is discrimination of phonetic contrast, kind of a classic question in the field. The second is statistical learning. And the third is spoken word recognition. I'm just going to go through a couple of studies just to illustrate the power of these behavioral methods. So in one paradigm called the head-turn preference procedure, infants are seated in their parents' lap, and they're presented with auditory stimuli that come from a loudspeaker, either on the left or on the right. And it's simply a measure of how long infants are willing to attend to certain kinds of auditory stimuli as indexed by their looking at a rather uninteresting blinking light. And this has been useful because you can precede the preference procedure by an exposure to a particular kind of stimulus and then ask whether infants can discriminate between the familiar stimulus and the novel stimulus during a test procedure. And this has been useful in terms of revealing an interesting process that goes on in phonetic discrimination over the first 12 months of life called perceptual narrowing. And this is work by Janet Worker and her colleagues showing that when infants from an English-speaking environment are presented with phonetic contrasts that come from a foreign language, that is a language that they have not been exposed to, you can see that at 6 and 7 months of age, infants from an English-speaking environment are quite readily able to discriminate these non-native phonetic contrasts. But over the course of the second 6 months of life, that ability to discriminate them declines. So that's the concept of perceptual narrowing. And in a control condition where infants were actually coming from a native Hindi or a native Salish environment, those infants were quite readily able to make that discrimination. So it really is an effect of early experience with that particular language. And this has sometimes been called a use it or lose it idea, in the sense that if you're not exposed to the language, even though you were able initially to make the discrimination, that you don't need to make the discrimination, and your system detunes itself to those contrasts. In the statistical learning domain, Jenny Safran, Lisa Newport, and I published a study a long time ago now on statistical learning in the auditory domain. And the basic question here was, how do infants segment a continuous stream of auditory events into coherent chunks, that is, auditory word forms? They're not meaningful entities yet, but they are stretches of speech that will become relevant to the learning of meanings. And in particular, the question here was, where does one word end and the next word begin? And a simple example of that would be a variety of utterances that the baby is exposed to, in which the statistical relations between the successive syllables in those utterances could potentially be extracted. So for example, the likelihood that bay is followed by b in this particular example is 100%. And the other examples of T-bay and Tulba and Yorba are less statistically coherent. And that's because the statistically coherent chunks are what we would call words in the language, and the less statistically coherent entities are word boundaries. That is the syllables that extend across a word boundary. And the question was whether 8-month-old infants were able to extract these statistical regularities after a brief period of exposure. And the exposure was something that we created using synthetic speech stimuli. I'm not sure you're going to be able to hear this very well, but I'll play it anyway. Many of you who have and so you can see that that's basically a series of concatenated syllables, but in fact, the underlying structure is very simple. It's simply 4 3-syllable chunks. There's no immediate repetitions of those chunks, and they're statistically coherent in that each syllable only appears in one of these chunks. So it's a very degenerate example, but it was designed specifically to determine whether statistical information alone was sufficient for infants to parse that stream of speech. And the answer was that they could. The test condition was to present them with a triple of syllables that was a statistically coherent entity, in this particular example, go, la, tu, and contrast that with a part word that is a statistically less coherent chunk, consisting of the last syllable of one word and the next 2 syllables of the next word. And that preference procedure was used to determine whether infants would listen differentially to words versus part words. And the answer was yes. They listened to the part words longer, as if they were the statistically less coherent triples, and showing a novelty preference, which is quite common in this literature. You can also ask whether or not this ability is specific to language materials, and the answer was no, when we created stimuli that consisted of entities from musical notes within an octave, organized into triples in the same logical structure. We found exactly the same effect, suggesting that this is a domain general ability and is not necessarily specific to language. So the third general topic within this domain of language development using behavioral techniques is spoken word recognition. And this has been done with many, many experiments. I just mentioned one. The idea is to present pictures of objects simultaneously on a computer screen, and then have one of those objects labeled by a sentence For example, where's the ball or where is the apple? And use eye gaze to determine whether infants will look at the item that matches the word that is spoken. In this looking while listening paradigm, which was developed most readily in Anne Fernald's lab at Stanford, here's an example of a study in which, with Dan Swingley, in which infants are presented with these two stimuli side by side on a screen. And when they're looking at the distractor, they move to the target because the target was the word that was spoken. If they're looking at the distractor and that is the word that is spoken, then they stay looking at the distractor. So this is a very simple technique that has been used in many, many studies to study spoken word recognition. And a number of years ago, Elke Bergelson was interested in whether or not the particular pairings of items that are presented on trials has an effect upon the likelihood that infants will look at the target word that is spoken. And in particular, she picked item pairs that had some semantic relationship with each other. And what she found is that even infants as young as 6 months of age will look significantly more at the named target when the competitor images, that is the other image that's presented on the screen, were semantically unrelated. So for example, they were more likely to look at foot when it was paired with milk than when it was paired with juice, indicating that even in the earliest phases of spoken word recognition in infancy, that there's some relationship or competition between the items that are represented in memory based on their semantic relatedness. And Elke also did a really interesting component of this study because she collected headcam data from infants in their natural environment in the home. And from these headcam data and audio recordings could determine the correlates of their performance in the laboratory. So in the laboratory, they were tested on these variety of item pairs. And you could ask, well, which of those item pairs did they perform better on in the lab based on the exposure that they had had in the naturalistic environment? And what Elke found was that the best predictor of word learning from the headcam data was the number of times that that word had been spoken while the infant was in fact looking at that particular object in their natural environment. So obviously this suggests that the kind of exposure that they get in their natural environment acts as a constraint on the kinds of word learning that happens very early in development. So this is a bit of a background on behavioral evidence. So how do we link brain and behavior? What can we infer about the brain from behavior alone? And of course, behavior is an existence proof that there's some minimally sufficient set of brain mechanisms that controls that behavior. And animal models are very useful in combination with studies of adults to provide detailed theories about brain organization and function that control behavior. But unfortunately, many of those invasive neuroscience techniques, of course, cannot be used with human infants. And many of the tasks that are used with adults are simply not the kinds of tasks that we can use with infants. So this leads to the potential benefits of using neural methods in studying these topics. And the three obvious candidates for neural methods that are potentially useful for infants are EEG, MEG, MRI, and NIRS. And so I'm going to talk a little bit about the pros and cons of each of these methods, because I think it's fair to say that there's no one particular neural method that is sufficient for studying any particular topic. So EEG obviously has excellent temporal resolution. It's relatively inexpensive, has relatively poor spatial resolution, and it's affected by some movement artifacts, particularly eye movements. MEG has excellent temporal resolution as well and better spatial resolution, but it's very expensive and it's subject to head movement artifacts. MRI is the gold standard in terms of spatial resolution, but it's very expensive, subject to head movement artifacts and relatively poor temporal resolution. And of course, NIRS has some combination of the two in terms of being moderately spatial and temporally resolving, relatively inexpensive, and not subject to terrible movement artifacts if it's used appropriately. In addition to these four methods, there's an exciting potential for optically pumped magnetometers, or OPMEG. Here's a recent review article by Brooks and colleagues. And this is using MEG techniques that are attached to a cap rather than having it be an instrument that the head must be aligned with. So similar to EEG and NIRS, I think this has some great potential in the future, but we're just in the early phases of implementing this with young children and infants. So the rationale for using these neuroimaging methods to study infant development, I think, is worth spending a few minutes talking about. There's really two, I think, competing inferences that we could make based on behavior alone. Imagine that we have a qualitative change in development, such as going from the ability not to walk to the ability to walk. That's a rather dramatic change in development. And it's quite seductive, I think, to think that that qualitative change is based on a new brain mechanism or a reorganization of existing mechanisms. But it's possible that it's exactly the same neural mechanism, but for some reason that neural mechanism was suppressed or affected by noise. The opposite is also the case. You could have the absence of a qualitative change in development. For example, in the statistical learning domain, there are relatively few developmental changes in statistical learning abilities, certainly not major qualitative shifts as in the case of walking. And so it's seductive to think, well, because there is the absence of this change, that it's the same brain mechanism that's operating across development. But it's possible that there is a new mechanism that replaces an old mechanism. And so the phenotype looks the same, but the underlying mechanism is different. Now, the only way to adjudicate between these two different competing inferences is to conduct neural studies to determine whether that neural mechanism, in fact, is undergoing change during development. And another important point about using neural methods is that behavioral development typically lags behind brain development. So if one of your goals is early diagnosis so that you can introduce an intervention or a treatment, then having a neural measure that you can see earlier than a behavioral measure could be really tremendously advantageous. So let's discuss now briefly how these neural methods have been used to study language development. And I'm going to go through fairly quickly a number of different questions within the domain of language development using these different techniques. So phonetic discrimination, statistical learning, spoken word recognition, and then a bit of work on language decoding and statistical learning, again, using fMRI and NIRS, and then finally measures of functional connectivity. So the classic ERP approach, as you're all aware, is to repeat a stimulus at random intervals and get an average evoked waveform. And this has been used to tremendous advantage in studies of infants. For example, looking at deviance after you have presented a repetitive stimulus to see whether or not they can discriminate that deviant from the standard. And this is just one example from Pat Kuhl's lab, where you can present native and non-native phonetic contrasts. And Pat and her colleagues have shown that this ERP component, the strength of that ERP component early in development at about 7 months of age, is a predictor of their subsequent word production several months later. Frequency tagging is another method that we're particularly interested in utilizing. And the basic idea here was illustrated in a study here by DeHering and Rossian, in which a stimulus is repeated at a fairly rapid rate, and a novel exemplar is introduced at a fixed number of repetitions of this stream of stimuli. So for example, here we have a case where every fifth stimulus is a face. The other stimuli are random. And so the question is, can you see a component in the steady state evoked response that is time locked to that 1/5 interval that is presenting this particular category, that is the face category. And the answer is yes, that component is present at that 1/5 frequency component, indicating that that stimulus has been discriminated from the background stimuli. And this has been used in the statistical learning domain out of Janet Worker's group to study the response to the same kinds of stimulus sequences that were used in the Safran experiments behaviorally. So to refresh your memory, you have a stream of syllables. They're organized into triples. And the question is, do you see a component in the steady state evoked response that is both at the rate of the individual syllable, but emerges as learning occurs at the rate of the triple? And the triple would be an indicator that they have extracted that coherent chunk from that stream of speech. And as you can see in the results from their experiment, you have the syllable frequency effect, which is quite robust, but you also have a reliable word frequency effect, indicating that this frequency tagging technique can extract the statistical learning process that has occurred in infants. Another technique that I think is extremely important, and one that we have been pursuing rather vigorously over the past 5 or 6 years, was articulated extremely nicely in Stan and King's paper in Ticks. And the basic idea there is to use EEG activity, not looking at single electrodes to look for an evoked response in a single electrode, but to look for the pattern of activity across all of the electrodes. And so the basic idea is to run machine learning algorithms at each time step in the signal across all of the electrodes and ask whether or not there's a pattern of activity in the EEG response that is correlated with particular stimulus. And what you would expect then is to be able to decode those stimuli as a function of the time post-stimulus. And moreover, you can train the algorithm at a particular time slice and ask whether or not it generalizes to other time slices. So we have used Laurie Bayet in my lab and Chuck Nelson's lab, she was a postdoc with the two of us, use this technique as outlined by King and DeHaan to study a visual categorization in infants. So we had a EGI 128 channel net. We used essentially 1 year old infants, and we had 8 visual stimuli. And the stimuli fell within 2 categories. So we could decode at the individual stimulus level as well as the category level. So we had animate and inanimate. We had animals and body parts as our 2 categories. And in adults, you get reliable decoding. You can see that the decoding begins post-stimulus at chance, as you would expect, and then rises to its peak at about 200 milliseconds post-stimulus onset. And in infants, you get a somewhat similar, although noisier, decoding function. The peak accuracy is not nearly as high as it is in adults. We think that's simply because infants are subject to a variety of noise factors, including inattention. But the point is that we can decode both at the individual exemplar level and at the category level. And as you know from your colleagues in Gislan's group, Julia Gennare's dissertation used these decoding techniques to study phonetic discrimination in 3-month-old infants using high-density EEG. And the impressive feature of that study, among others, was the ability to decode these stimuli even when they were spoken by different individuals, and even when they were created in syllables that varied in their vowel structure. So both manner and place of articulation could be reliably decoded from EEG. Now we've taken this decoding approach and extended it to the level of the lexicon. So moving up from phonemes to words. This is work with Bob McMurray at Iowa. And what we wanted to determine is whether on a millisecond by millisecond basis, as the speech stimulus is unfolding, could we decode the particular word that was being spoken and other words that were acoustically similar in the lexicon to that word that was being spoken. And so let me give you a little bit of background on why that's particularly important with regard to what we already knew from behavior. So many of you may be familiar with the visual world paradigm. It's a somewhat more elaborated version of what I referred to earlier. So instead of having an apple and a ball, and you are hearing one of those two words, you see four objects on the screen simultaneously. One of those objects is the target, in this particular case, bug, and the others are related to the target by acoustic similarity, as in the case of bus, because it begins with the same set of phonemes, and two of the other stimuli are unrelated. And the basic idea is that over a course of many trials, as you present one target word in the context of these four pictures, you're going to have eye movements that go to, that is, are directed to the target word. But as you can see on these examples in trials 3, 4, and 5, there's some errors that are made in those eye movements, at least initially. Sometimes they go to the competitor word and then correct and go back to the target. And sometimes they go to the unrelated, although that's very rare. So what you can do across a series of trials is integrate the probability of looking at each of those four stimuli as a function of time. And that's illustrated here schematically. So you can see that the target gaze probability rises very rapidly and is quite reliable. The competitor activation, the onset similarity competitor, shows a very similar rise, but then as more acoustic information comes in, that declines, because the likelihood that that spoken word matches the target picture is low, right? Bus does not match the picture of bug. So this has resulted in lots and lots of behavioral studies using the visual world paradigm, and these are actual data from one such study showing that very same pattern. Very reliable gaze directed to the target word, some activation or gaze directed to the cohort or the competitor word, and even cases where the rhyming word, where the end of the word is similar to the target word, can be detected. This has also been done, as I mentioned earlier, in infants and toddlers, and it's also been done to measure aspects of spoken word recognition that have to do with disfluencies in how we speak. So what we were attempting to do was to use EEG to obtain a neural measure of this lexical activation and competition. And the basic idea is very simple, similar to King and DeHaan. We're taking a set of electrodes, we're measuring the activity from those electrodes as a pattern, and we're looking at that pattern of electrical activity at each time step as the spoken word is unfolding. And using a support vector machine algorithm, we're looking for a pattern of EEG activity that can reliably discriminate or identify one of the 8 words or one of the 8 non-words that we used in this particular study. And for those of you who are into the machine learning techniques, it's 15-fold cross-validation. So we're withholding a set of trials to train the model and then test on those withheld data. And we actually use 2 different EEG systems, both a gel-based system and the saline-based system, the EGI system, because we wanted to determine whether it was reliable with both. So this immediately raises, in the decoding world, how do you determine which features of the EEG you're going to use to seek a pattern of activity that is correlated with a stimulus? And for those of you in the EEG world, there are many different features that you could imagine using. In the Laurie Bayet study, we used simply a low-pass frequency raw EEG signal. That's all. It was very simple. You can also look at oscillatory components, alpha, beta, theta activity. You can look at the ERP waveform at each time slice. And of course, you can look at combinations of these features. So what we did is we took 5 adult subjects, we collected data from them, and we looked at all possible features that we could think of that might be relevant to this decoding. And it turns out that the ERP waveform was the most reliable component. And in fact, the oscillatory components did not add any additional significant decoding accuracy, which was quite surprising to us. So nevertheless, the idea is that at each time slice, you're getting an ERP component, and you can characterize that component by, an underlying function, and you can look at different size temporal windows that define that ERP component. And you can then do that, for example, at every 20 millisecond time slice. And so that's what we did. Once we had frozen for those 5 subjects that we did this analysis on, the features that we were using, we never changed the features again. We just deployed those same features across all the other subjects that we gathered data from. Now, the design of the experiment was also very simple. We had a set of words that had similar onsets. So you can see badger and baggage. And we had a set of non-words that also had similar onsets, bab, bib, and bad dough. And we simply had subjects listen to these words repeatedly in random order. There was no task. And then after this passive listening, we did a delayed verification task. So for example, they'd hear the word badger, then there would be a pause because we wanted the EEG activity to be unrelated to the motor response. And then they had to verify by looking at a text version of the word, whether it was badger or baggage, and they made a motor response. So what we're doing then is we're asking whether or not we can decode on a trial by trial basis and a millisecond by millisecond basis which of several possible target words are being spoken at that time. And what you would expect from the behavioral data is that initially, when you only have access to the first set of phonemes, both of these words have the same set of phonemes. So both of those word candidates should show activation in the EEG activity. But then with further acoustic input, as the target word is clarified by further acoustic input, the competitor words activation should decline and the target words activation should reach an asymptote. And that's exactly what we found. So in the EEG decoding data, we see essentially a mirror image of exactly what we found behaviorally in the visual world paradigm. Interestingly enough, it was present both for words and for non-words, and we did not see significant differences between the activation for words and non-words in terms of overall decoding accuracy. This suggests that this technique at this stage is useful primarily as a measure of the acoustic phonetic information and not the word meaning information. Otherwise, we would have seen differences between words and non-words. But importantly, we can gather sufficient data from individual adults to see highly reliable activation functions for each individual. And we're particularly excited about this because we're engaged in a colleague of ours, Liz Simmons, and other collaborators of ours to decode individual words and their cohorts for children. And this is a study that's ongoing of children who are late talkers. That is, they don't speak very much. And the question has always been in late talkers, is that because they have a word comprehension problem? And this is a way to measure word comprehension using both the EEG decoding technique and an eye tracking measure. So we're going to do both behavioral and EEG studies. And the contrasts are quite simple, right? They're simple for children. This is an example of kitty versus kitchen, and that would be one particular pair of words. So I think in terms of summarizing the decoding work that we've been doing and others have been doing, we have to ask ourselves, what does decoding tell us? It tells us that there's some pattern in the brain activity that's minimally sufficient to predict a behavioral state or a stimulus that's being presented. And it's a powerful tool, we think, particularly for preverbal infants, because they don't have to make a motor response. But is that pattern a mechanism? I don't think so. It allows us to look at a pattern of activity and then to try using other techniques to make inferences about where that pattern of activity exists in the brain and what underlying network is involved in that pattern of activity. But it's not a direct readout of that mechanism by any means. But I think it's a metric like behavior. It's like percent correct. And I'm totally fine with that in many applications, even though we don't necessarily understand the exact neural mechanism. If we can have a reliable neural measure, I think that that's an important goal in and of itself. So I wanted to spend just a little bit more time talking about the two other techniques that have been used, the hemodynamic techniques, near-infrared spectroscopy and fMRI. And I'm sure many of you are familiar with fNIRS. It's a diffuse optical imaging technique, measures both oxy and deoxyhemoglobin, and you can present an array of emitters and detectors on a cap on the baby and record from different locations in the surface of the cortex. And a classic approach in using NIRS came out of the group that I'm speaking to now with Jacques Mehler, in which forward and backward speech was presented to newborn infants and showed canonical left temporal cortex activations to that contrast. But that's a univariate approach. You're looking for particular ROIs that are activated. And another useful technique is a multivariate approach, in which you're looking at the pattern of activation, like we just described in the case of EEG, but now in the case of fNIRS across a series of channels that are collected from the head of a particular subject. This is work by Ben Zinzer and colleagues. Again, using those very same stimuli. These happen to be visual stimuli and auditory stimuli together, so they're pictures and words spoken at the same time. And what's interesting about this particular instantiation of a multivariate approach is that you can gather group data and ask whether each individual subject's decoding accuracy conforms to the group average, which, as you can see on the left, it does. But you can also decode the performance from each individual subject based on rating data of semantic similarity obtained from adults in corpus analyses. So the multivariate technique has the advantage that the decoding can be done both based on the signals and based on computational models of how those signals should be related to each other. And we've used this technique. Lauren Emerson and colleagues have used this technique to do a multivariate analysis on infant NIRS. And the idea is very, very simple. Imagine you just have two categories of stimuli. One is an auditory visual pattern, and the other is a different auditory visual pattern. And the question is, can you decode from the NIRS signals in infants whether the infant on that trial is hearing pattern number one or pattern number two? So there's just two stimulus conditions. Now, the difficulty with fNIRS with infants is that it's difficult to get enough training data on each infant to do decoding at the infant level. So the strategy that we've used is to have a leave one infant out design, in which you use all the data from all the infants except one as your training data, and then you test it on each individual infant that's been left out from the training data. And even with this very, very simple binary classification, using just a relatively small number of NIRS channels, we were able to get decoding accuracy of 72% correct. So it has the potential of being a very useful technique, the multivariate technique with infants using NIRS. Now, the classic fMRI approach, again, from your group, it was instantiated in a study that, again, used forward versus backward speech with 2-month-old babies, and again was an ROI approach showing that there were left hemisphere activations to this stimulus contrast. Subsequent work from several different labs, including Rebecca Sachs, looking at faces versus scenes, kind of a classic ventral temporal activation question, has shown very similar patterns of activation in infants and adults to this contrast between faces and scenes, suggesting that the basic architecture for both speech and vision is already set up in quite young infants. One of the difficulties associated with fMRI, of course, has been whether you can do it with awake babies. Now, obviously, the visual experiments have to be done with awake babies. And my colleague, Nick Tork-Brown at Yale, has been spending the last 5 years trying to get this fMRI technique to work well with awake babies with quite great success. This is just an example of each individual baby. And basically, the average data that you can get, good quality data that you can get out of each individual infant is about 10 minutes. So Nick has done a number of studies using this awake behaving fMRI technique, one of which is a visual statistical learning study. This is Cameron Ellis's work. Cameron was a postdoc in my lab for a year, showing that there's hippocampal activation, which is obviously you could not obtain hippocampal activation data from NIRS because you can't measure deep brain structures, and showing that over the course of statistical learning, there's an increase in hippocampal responsiveness that's correlated with statistical learning. Another technique that can be used with fMRI to great effect is functional connectivity. And you can look at large-scale brain networks, compute each individual pairwise connectivity across different parcels of the brain, and then measure the relationship between the connectivity patterns and some phenotype. And Marvin Chun and Todd Constable and their colleagues at Yale have used this in a variety of tasks, including measures of attention, IQ, and psychopathology. So any phenotype could potentially have a unique connectome-based organization that's correlated with that phenotype. Now, measuring functional connectivity using NIRS has been going on for about a decade now. Most of it has been done with sleeping babies, and most of it, therefore, has not been related to their phenotype, right? It's been simply looked at correlations with age, for example. But an interesting example of this in the fMRI world that I think we could use with great utility in studying infants is a study out of Stanford in which sleeping infants had their functional connectivity measured in the scanner. And then data were collected in the home while infants were interacting with their parents. And the Lena recordings in the home measured how many turn takes were there between the parents' interaction with the baby and the baby's attention and utterances. And then what they did is they looked at functional connectivity between different networks in the brain and asked which of those networks were correlated with the data that had been collected in the home, the behavioral data in the home, and found that these turn-taking, bouts of turn-taking in the home were correlated with this posterior temporal network. So the point here is just an interesting example of the relationship between a behavioral measure in the natural environment and the connectivity that you can measure in the resting state in fMRI. So I hope I can take 5 more minutes, Jessica, to go through some what I think are interesting applications of naturalistic viewing paradigms with neural methods. So we talked about resting state networks, and I think it's important to make a distinction between resting state networks and task or awake behaving networks, because they do differ, and how these properties change over development. And then make a distinction between what the decoding models that I described earlier and encoding models. So that's where I'll end. So the power of naturalistic task, I think, has been made very clear by Uri Hassan and his colleagues. There's a nice review paper here. And functional connectivity can not only be measured within individual subjects, but can also be measured between subjects. And I'll get to that at the very end. So Sarah Sanchez-Alonso in my lab has examined functional connectivity relations in a large data set from the Healthy Brain Network. These are fMRI data. And what has shown quite convincing evidence that the pattern of activity in the networks in the brain differ very greatly between watching a movie and resting state. So the notion that the resting state is a sufficient characterization of the brain, I think, is just incorrect. It's different than it is during active engagement with the task. Moreover, you can decode whether an individual subject is in the resting state or in the movie watching state with great accuracy, even with only 3 minutes of data. Moreover, Sarah showed that difference between resting state and the movie watching state, that itself changes with age within the language network, suggesting that there are significant changes in the language network over the course of development, which is not surprising. We've been very interested in using NIRS to get at the movie watching functional connectivity networks. And we have an ongoing study with a preliminary version in which infants in English-speaking environment are listening to short movie clips in which the audio track is either English, Spanish, or a distorted version of speech. And we can see highly reliable differences between the activation in this functional connectivity network between English and Spanish, between their native language and a non-native language. And moreover, when we look at either seed-based connectivity or all possible pairwise connectivity relationships in the fNIRS channels, we see reliable differences between listening to English and listening to Spanish. So we are interested in using these functional connectivity data to decode whether the infant is listening to English versus Spanish. We haven't done that yet. I think I'm running out of time, Jessica, so I'm going to just go through this really, really briefly. We've been talking about...
Please take your time.
We've been talking mostly about decoding models, and I think it's important to make a distinction between a decoding and an encoding model. In a decoding model, what you're doing is you're taking the EEG response, for example, or the NIRS response, and you're asking whether or not you can predict some particular stimulus feature. For example, the phonetic quality of that stimulus. In an encoding approach, you're doing the opposite. You're taking the actual stimulus features and asking whether or not you can predict the EEG response or the NIRS response. And you're doing that by looking for a temporal response function. So this technique is powerful because you can look for different stimulus properties, for example, the phonetic information, the acoustic waveform, word frequency, or the semantics, and use each of those as your predictor of the EEG response. So you can parse the EEG response at multiple levels of the language hierarchy. And this has been done with NIRS, in addition to fMRI, out of the WashU group, Joe Culver, where they're watching a movie, and what you can do is decompose that movie into its underlying components across time. For example, the auditory features of the movie or the visual features of the movie, and then ask which of those pairwise feature correlations is predicted in the fNIRS activity in different locations in the brain. And that's exactly what Joe and his colleagues had done with adults. And it's also been done in a preliminary way with infants, Jessen and colleagues with 7-month-olds, where they're watching a cartoon. And just at a very simple level, they're identifying the audio components, the motion components, and the luminance components of the movie, and then seeking this temporal response function that can predict the EEG response. And the interesting thing about this particular study is that temporal response function differed between infants and adults, suggesting that the underlying way in which the brain is organized to process these different levels of linguistic information and visual information differs considerably with development. And finally, this is my last slide. I just wanted to mention that this approach, the encoding approach, can also be used in social interaction. And this is a preliminary study, I think very interesting, of Elise Piazza's work at Princeton, where mothers and infants are wearing NIRS caps and interacting with each other. And you can look at the interbrain functional correlation matrices as a function of time. Now, what they did not do, but what they could do using an encoding model, is that they could look at mutual gaze and maternal speech and ask whether or not those regressors have a temporal response function that is indicative of the kind of interaction that goes on between the members of the dyad. So I think has a lot of potential to use this encoding approach. So by way of summary, let me just quickly finish up. These behavioral studies that I mentioned early on of infant language development have a long and productive history. Studies of neural development add an important insight about the mechanisms that mediate these behavioral changes. The multivariate approaches are quite mainstream in adult neuroscience, cognitive neuroscience, but are just now being deployed with human infants. And I think a variety of practical constraints have held us back in studying infants using these neural tools because we need a lot of data. And the naturalistic viewing task is a way to get a lot of data from individual infants so that we can have more reliable indicators. And that will be, I think, particularly powerful if you're studying individual differences, because these techniques allow you to get enough data that you can say something about individual subjects. So with that, let me conclude, and I'm happy to address questions. For those of you who are watching this on Zoom, on video later, feel free to email me if you have any questions, but I'm happy to take questions live now.